Good afternoon, everyone. Can you hear me?
Yes, ma'am.
Yes, ma'am.
Okay, okay,
Yes,
thank you.
ma'am.
Good afternoon, everyone. Can you hear me? Am I audible to everyone?
Yes, ma'am. Yes.
Okay,
Yes, ma'am.
thank you. Uh,
Yes, ma'am.
good morning, respected resource person, Dr. Payal Chakraborty, ma'am.
Thank you.
Uh, already one resource person joined with us in this online workshop. So once Principal Father from Holy Cross College, Agartala will join the session, we'll start our today's program. Thank you once again. Good afternoon, everyone.
Good
So
afternoon.
today, our resource person and Dr. Shovik Ghatak and Dr. Payal Chakraborty ma'am is with us right now. And in the meantime, our principal father, Father Dr. Benike John CAC, already joined with us. So with due permission, from father, I'd like to request Dr. Ashish Kumar Sinha sir, Assistant Professor, Department of Human Physiology, to give a brief introduction regarding this today's online cancer awareness program. Over to Ashish sir.
Thank you, madam. Am I audible, madam?
Yes, sir, you are audible.
Okay. So, good afternoon, everyone. I am Dr. Ashish Kumar Singh. On behalf of the organizing team, welcoming all of you in the workshop entitled One Day International Level Cancer Awareness Workshop. In this workshop, in total, 453 number of total participants are registered. Uh, and so far I mean we have now joined around 68, but hopefully during the progress of that this meeting, I mean workshop, they will all join. And if it I mean more than 100 participants, we will also just share the link, that is YouTube, for the YouTube live. The target audience for this workshop is basically undergraduate and postgraduate student, so that they can aware the factualities and realities about the disease cancer and its prevention. Our research persons are renowned researchers having vast experience on cancer research in their uh, research career. I hope we are going to learn recent advances and new aspects on cancer from this workshop. Now I would like to invite our respected father, Dr. Benny K. John, CSC, Principal, Holy Cross College, Agartala, to grace our workshop with his precious words as a welcome address. Father.
Thank you, Sir Rajesh. Uh, Dr. Payal, Dr. Shobik, faculty
Hello.
members, college and dear students, a very good afternoon. I am privileged and to be happy to be with you for this webinar. The very topic itself on the cancer awareness workshop speaks a lot about the significance of this topic and the large number of scholars and students that you have registered also is an indication that how important and how much we are interested to get more and more to become aware of this topic and how we can be also maybe messengers and agents to alleviate and prevent or support the all effort to a cure or to support those who suffer from this illness, also to prevent ourselves or protect ourselves also from possible ways and means that will be maybe we would learn from our resource persons. At the very outset, I would take this opportunity to congratulate the science department, human physiology, and other departments who have joined in hands together to organize this wonderful webinar. And on our part, from administration and from the management of this institution, Dr. Payal and Dr. Shobik, we are very grateful for your time, your talents, and your goodwill to spend time with us and share your knowledge. And we are honored. And once again, I welcome everyone warmly to the webinar. And I too am going to be with you and look forward to have a good time. Good day, and thank you.
Thank you.
Thank you, Father, for your inspiring words. Now, I would like to hand over the session to Dr. Susmita Saha, Head of the Department and Assistant Professor of Department of Physiology. Over to you, Madam.
Thank you, sir. Thank you so much. Actually, today, first of all, I'd like to share one thing. As sir mentioned, today, total 453 participants registered themselves for this to attend this workshop. So, here in this platform, Google Meet platform, only 82 participants joined here. So, simultaneously, we'll share the YouTube link as well as the Facebook live link to the registered email ID so that everyone can attend this online workshop. So with this, without any delay, I would like to request Dr. Somnath Kaur sir, Assistant Professor, Department of Botany, Holy Cross College, Agartala, India, to introduce our first resource person, Dr. Payal Chakraborty ma'am. So just to deliver her lecture in this online platform. Over to Somnath sir.
Thank you, madam. Madam, am I audible?
Yes, sir, you are
Am
audible.
I audible, madam? Good afternoon, respected Dr. Father Benike John, CSC, Agartala, Father Joe Paul, CSC, Vice Principal, Holy Cross College, my esteemed speakers, respected faculty members, and my dear students. It's my privilege to introduce Dr. Payal Chakraborty, a distinguished researcher in the field of biotechnology, whose work is shedding light on the intricate genetic landscape of gastric adenocarcinoma. Dr. Chakraborty's academic journey has been marked by unwavering dedication to scientific exploration. With a PhD in biotechnology from Mizoram University, Aizawl, India, her research focus centers on the identification of recurrent genomic alteration of gastric adenocarcinoma in the Mizo population. Her work in the area has been potential to significantly enhance our understanding of the genetic factors underpinning gastric cancer. Dr. Chakraborty's academic prowess is evident in her honors degree and MSc degree in biotechnology from Bardhaman University and Behrampur University, respectively. These academic achievements provide the foundation for her groundbreaking research endeavors. Her contribution to the field has garnered recognition throughout the prestigious award and the grant, including the biotechnological career advancement and reorientation, that is BioCare, Women Scientist Award from the Department of Biotechnology DVT, and a research grant from the Royal Physiographic Society of Lund, Sweden. Dr. Chakraborty's research journey has transcended borders, taking her to the Lund University in Sweden, where she presently serves as a postdoctoral fellow. Here she is actively engaged in exploration of WNT and beta-catenin signaling in colon cancer, employing a range of advanced molecular and cellular techniques. Her unwavering commitment to the scientific inquiry to exemplified by her extensive list of publications in many national and international journals. This publication underscores her contribution to advancing our comprehension of the complex interplay between genetic and environmental factors in the context of gastric cancer. So now I request Dr. Chakraborty to deliver her presentation. Over to you, ma'am.
Thank you very much for this wonderful introduction. I'm overwhelmed, and I'm a girl from Tripura only, so it's a pleasure for me for getting this opportunity to deliver the students here. So I am very grateful to Holy Cross College and all of you. So I think I will try to share my knowledge or experience with the students. So let's hope they will be beneficial. Is my slide visible?
Yes, ma'am, it is visible.
Okay, thank you.
Yes, ma'am.
Okay, so today I'm going to deliver I want to discuss about the cancer scenario and as well as awareness and the risk factors which are responsible for the cancer uh, in worldwide as well as in our country. So I'm at present I'm working as a postdoctoral scientist in Lund University here after I have completed my PhD actually in 2021, and then I got a chance for continue my postdoctoral research in this Lund University. So yeah, now I got a award also from DVT. So next I'm planning to do something in India for our country if I can benefit with my experience to enhance to know the cancer research in future also. So to know what is actually the scenario of cancer, first we need to understand in worldwide also. So can we, as we can see that it's nearly about 18.1 million new cases are there already registered in 2020, which is a very huge case, I think. And then in case of mortality also, it is very high. It's 10 million deaths in the year 2020 only. So it's like a 1 in 6 deaths average. So we can understand that it's a very huge problem. So it's a very lethal disease which is continuously overgrowing, and we need to go somewhere to overcome the problems. So if we see here, we can see that the most of the part of Asia, this is where we are also belongs, like 49.3% of Asian is a hotspot for cancer developing. So we need to actually inquire why it is happening in Asia, not in as Asia is also a developing country. I mean, so many developing countries there in Asia. But developed country, already the Western countries are having less compared to Asia. So yeah, it's a matter of thinking. Coming to now in India, so as we can see that Nearly about 1.4 million cancer cases are there in 2022 itself. And then from these statistics, we can say that so 1 in 9 people are likely to develop cancer in his or her lifetime. It's a statistics. So in case of death or mortality, it is going to be means it's like a estimating thing. Then it's maybe cancer burden will be 12.8% in 2025 as compared to 2020. And coming to the like recent PBCR report, so they have told that the cancer burden will be 29.8 million also around 2025. So if we see here, it's a statistics for India only in male and female. So in male, we are seeing that lung cancer is the highest topmost cancer, while in female it is breast cancer. So we'll try to focus like this top 5 cancers, which are include among them the stomach cancer is also here, which I have done PhD in my PhD in stomach cancer. So in case of these are the top 5 cancer in males and females. So it's obviously breast cancer is a very obvious threat for females, and then lung cancer is a topmost threat for males, including other like mouth, prostate, tongue, and stomach. And in case of female, it is breast, cervix. Of course, cervix also another cancer which we need to think. And then ovary, then corpus luteum, and lung cancer. So now I want to show the scenario in India where the cancer is very prevalent. So as we can see that our northeast region, so we are belongs to northeast region, and it is a very highly danger hotspot for developing cancer. So we are at very utmost risk. We are like a risk group for developing cancer in India, but it's a matter of really thinking. Let's see what we can do. Miss, I will try in this lecture. I will try to make you aware about the cancer, means why it is happening and why it is more in northeastern region, or maybe why. How can we solve this problem? So if you see that the highest cancer incidence rates in northeast India, the states are like Mizoram, Arunachal Pradesh, and Nagaland. So hopefully actually it's, I am happy that Tripura is not here till now, but it's maybe it can be there in this list anytime. So as we can see here, like Tripura in our state, the highest the averages in Kamrup urban districts, where in for one one is infected with cancer in maybe in 4%, but in Tripura also it's there, like it's in within 11%, maybe one is having cancer. So yeah, it's a very alarming position for our state also. So in case of like females and male statistics, what are the top most common cancers in northeast region? And they are like, for female it's like breast, cervix, gallbladder, esophagus, and lung. And case of males, it is esophagus, lung, stomach, and follow through this hypohearing and mouth. So actually, when I search for which is the highest cancer is now at present in Tripura, it's esophagus. So yeah, we need to go through for esophagus cancer also, why it is happening in particular this state. So now actually I want to say very basic thing about cancer. First, we need to know what is cancer, means why it is different from normal cells. So as we can, it's like a in book chapters also, in our classes also, we used to read that like normal cells, if you can see that it's very organized, boundaries are very organized, nucleus is impact, cytoplasm is their space for cytoplasm is small, and chromatins are also nicely organized here in the normal cells. But when it's come to come to cancer, So there you can see that its nucleus can be more than one also, and chromatin you cannot differentiate properly, and the boundaries are also like very disorganized. It's like you cannot differ also sometimes. What are the is it two cell or three cells? Means the you can see the structural differences between the normal and cancer cells. So coming to the like if you see the stay in the human cells, then you can see that so it's like epithelial cells or epithelial cells. So here in normal, they are so much organized, they are well designed, so we can separate all the boundaries of each cell in our human tissue. But when it's come to tumor, you can differentiate it. So there all the cell boundaries already distracted. There is nothing to see. Means you cannot clearly see the boundaries. You cannot differentiate between the cells. What is happening? These are already cell membranes are distracted. So nucleus are here and there. Means it's a totally it's an advanced stage of cancer. So now coming to the stage kind. As you can see, normal epithelial cells, these are the orange one, are the normal epithelial cells. It's nicely organized. But then when it's going to develop cancer cells, its nucleus is coming bigger and cytoplasm is having very less place. Like in T0 stage, it's like a development, just it started cancer developing. It's not yet harmful, but it started. Then in case of stage 1, as we can see that, it's going to be mucosal level. It's now start invading the mucosal level. And the size of the cancer cells is very much bigger than the normal cells. And coming to the stage 2, like it's a T2 stage, and there you can see that it's started submucosa, mucosa level also, and then it's going to up also. So it's invading more. So the case or disease is getting more worse. So in case of stage 3, you can see it's almost invading the all tissues. It's coming to be invade the outer layer also. So this is the situation in stage 3. And in stage 4, it's already out of the tissue, and it's in the now it's like in stage 4 it's like now it's in the bloodstream. Then if it is in blood vessels, through blood vessels, the cells used to carry in the other organs, then it's called a metastasis. If your cancer cells is going to carry to the other organs, then you can say that it's metastasis, and it's a distant metastasis. That means the cancer is very much smart, and their growth is like in another level compared to the normal cells, which is not good for us. So anyhow, we need to think the way how to decline the growth or how to make them apoptotic. So this is like a introduction for cancer. It's basic idea. So now we have to think what is the cause behind cancer. So in my lecture, I will try to aware you about the causes why we are getting cancer and the risk factors. So if you can see the image which I am showing just now, cancer actually is a complex disease. It's not a single factor which is associated with the developing for cancer. It's a multifactorial disease, means like genetic causes are there, environmental factors are there, which are the risk factors for developing cancer. If you can see this, it's a train track line. So it's like it is the situation. What are the causes of developing cancer? It may be your age, it may be your sex, it may be your food habits, it may be your dietary habits, but all together it's connected. One environmental factor or one dietary habit is affecting the other factor. That's why it is too complex to target only one region or only one gene in case of therapy. We cannot target one gene or we cannot target one signal because they all are interlinked. So that's why it's quite tough to target one gene or one protein or even to target why it is happening. It's a network of risk factors. So, like, if you see what can be the risk factors associated with gastric cancer, so maybe it's I have written gastric cancer. It's for all the cancer, not only for the gastric cancer. So, like race and ethnicity, then geography, then age, gender, diet, lifestyle, habit, obesity, and obviously the pathogens like bacteria and virus. And we individuals sometimes we don't know, but we are a risk factor also. And how? I'm going to tell you later. And then family history and genetics, yeah. Then genetic alterations. Now I will discuss with one each factors to make you understand how. So coming to race and ethnicity, as we all know that worldwide we have different race and different ethnicity. So some are black and some are white. So if you see that means prediction of cancer rates and outcomes, always it's differ to population. Even in India ourselves, we are having so many populations, so many ethnic tribe groups. So they all are having their own ethnicity. So it's the cause behind the cancer in different population is also different. It's vary from population to population. So we can have like a population specific mutation or population specific polymorphism, which is or which will be responsible for later stage for developing cancer in this group. So like yeah, every population and race has their unique dietary habit and lifestyle habits, which is also affecting the alterations. And coming to genetic factors, and yeah, one more important thing is socioeconomic background. So like that also means in our society, we are not belongs to from the same socioeconomic background. Some is very rich, some is poor. So that's why to avail facilities or to afford quality care, it's not happening all time the same. Healthcare access also, it's differ population to population and ethnicity to ethnicity. So this is also a cause. So if in Western countries, if you see that the white women are slightly more likely, they are more at the risk group compared to the black and Hispanic and Asian women to develop breast cancer. But the black woman, whenever it's happening in the black woman, these cancers are more aggressive. More aggressive means this can be triple negative. Triple negative means if in your breast cancer, it is the most important hormones are like estrogen, progesterone, and HER2. If these three are coming negative in your breast cancer after screening, then we cannot go for any therapy for that patient. So that is more aggressive case. So which is happening in black people compared to white people more. So as I have done my PhD in Mizo ethnic group, so in Mizoram, so they are also having their unique food habits, lifestyles, and they basically used to practice endogamy. What is the meaning is that they used to marriage between the tribes, means not going for other tribes or not going for other caste between the caste. So this is also a cause for developing cancer. Coming to the next, like geographical region, yeah, then how does it matter? Geographical region also matters, and how? So like yeah, different populations are in different part of world, and they are also responsible. Means in some part of world, this one cancer is very predominant, but in other that may be other cancer. So basically, like in cancer is really prevalent and in the eastern and central part of Asia. So if I want to give you one example, like how geographical region is going to affect. So it's like uh, sun, a different geographical region is having different uh, sunshine also. Means somewhere it is very less and somewhere it is very strong. So these are the factors which is relating with the skin cancer. So yeah, if the sun is very less, you are very less exposed to sun sometimes, then it is you will having the vitamin D deficiency and you will get a skin cancer. You will get a chance of skin cancer in future. And then if you are overexposed, then also it's a matter of risk factor. So it should be, that's why some geographical region is also matter for developing cancer. Next, coming to age, obviously the older patients or older people are the group which are more prone to developing cancer. Well, basically, if you see the cancer patient's age, it's basically you can see that about 50 means before the lower level limit. After 50 it started, and we are having patients more number than after the age level of like 50, 60. So why? Means we can tell that, yeah, as your life span is increasing, your age is you are going to older, you will be more exposed to like UV radiation or maybe harmful chemicals, or maybe your lifestyle means your lifestyle habits like tobacco smoking or alcohol drinking, or maybe some viruses can expose you more. So that's why maybe we are getting more older patients for developing cancer. And another thing, it can be immune system also. Our means if we are getting old, then our immune system obviously going to decline. So that's why the old age is a matter of concern. So coming to gender, yeah, we have some gender issue also in case of developing cancers. So besides some gender specific cancer like prostate cancer for men and breast cancer for women, but nowadays we are having breast cancer in male also, which is quite surprising. But then cervical cancer for female. Besides those cancer, but if we can see, we are having actually most significant, sex is becoming most significant factors because men are more likely to develop cancer than women. It's we can see for every cancer we are seeing a statistical trend that men are more prone to get women. So why? Maybe so actually if you see the statistics, it's showing you that in 1 in 2 chances being diagnosed with cancer during their lifetime for men, but in case of female it is like a 1 in 3. So yeah, obviously gender males are in a more prone risk group to get cancer. So I will come to diet, but I want to say something about the gender again. So maybe male persons are more exposed to lifestyle habits like smoking, alcohol drinking, tobacco chewing. That's why maybe they are getting more and more genetic alterations in their gene, and they are getting more cancer. So another important factor is diet. First, I will discuss about which type of diet we should exclude. So the main factor for any cancer, it's like extra salt. We should reduce our salt consumption. So actually, salt can make your make your gut more favorable for pathogen infection also, like H. pylori. It can make more acidic also. So we need to avoid salt. Coming to the refined sugar, means if it is better to get to raw sugar, or means like jaggery or honey we can use, but not the refined sugar. It is also a very risk factor to promoting cancer. So coming again, it is red meat like pork, then mutton, then beef. It's also going to be a very hazardous for, means it's going to develop the chemicals which are responsible for developing cancers in future. So we need to avoid these foods. Like smoked food, yeah, in smoked food we have the chemicals like benzopirin and all, but smoked foods like it can be smoked meat, it can be smoked vegetables. We need to avoid, because due to smoke there are benzopirin, they are generating benzopirin, which is very a harmful chemical for developing cancer. So we need to avoid again the fat food like dairy products also. So uh, with the food which contain fat, that should be reduced, and better to avoid the dairy products like uh, dairy products, and also like if it's good if you can avoid cake, chocolates, and everything. So coming to here, when I did my research with mesoethnic group, I find it they are having a they used to call it sound, like it's a fermented pork fat. So they are making in home. So in this sound bar, they used to preserve the pork fat for a long time, and they used to continuously consume in their regular food with this pork fat, which is very dangerous. So it's like it can make the favorable situation for pathogens infection, and which they are going in alter, they are going to make a adverse situation for our cancer developing. So coming to now, which we should include in our food diet. So this is like fresh vegetables. Yeah, we need to eat more and more vegetables, especially like broccoli, cauliflower, cabbage, and sprouts. Then which contains sulforaphane, which is having anti-cancer properties. So we need to focus to, means we need to include in our diet these fresh vegetables, and also fiber-rich fruits like rich in fiber, we can include in our diets. So here I have told only about apples, pears, and blueberry, but you can also add like banana and all. So coming to pulses, yeah, pulses like such as beans and lentils are always good to have in your diet, in your diet, instead of this smoked meat, red meat. You should consume more and more pulses. And whole grains, means don't go for the like which are flour that which whole grains is always good, not go for the factory, means the refined grains. It's good to go for whole grains. So if we follow this diet, so it maybe reduce our cancer risk. So lifestyle habits, yeah, we are having people, everyone is having their different lifestyles. So which we should not do. These are like chewing tobacco, which is a very common practice. Everyone maybe they are having a khaini, gutka kind of things. So which is a very common case for, it's a risk factor for developing cancer. And then yeah, pan with bitter nuts, which is a very common practice in Tripura. In every home, there are pan with bitter nuts. And my mom also very much fond of. They will not never listen. Then smoking. Yeah, smoking is another lifestyle habits. You should avoid smoking. It is very injurious to health. And alcohol also. Coming, then again, it's a unique lifestyle habits like tuibu. Tuibu is a drink, means is a drink tobacco infused water, which specially we can found in Mizoram, and this is very carcinogenic. We have tested in our lab also. It's having all the chemicals which can make your condition adverse for developing cancer. So yeah, we should avoid these things. And in research it has came also that night shifts. So if we can avoid night shifts, then it will make you more stress. Stress is another risk factor which can uh, in future you can develop cancer if you are having more stress. So better to avoid night shifts. So which we have, so yeah, it's a tuber, like why it is. So it is having polyaromatic hydrocarbons and carbonyl compounds. That's why it's making, it's a risk factor for developing cancer. So these are the don'ts, and which we have to do. This is exercise. We must have to do the exercise daily or workout. It's maybe running, it's maybe walking, it's maybe your workout, whatever. But you should do always exercise. In this context, I want to say that in Sweden, I have seen that everyone is doing exercise. Means even the old age people like 80 or 90 ages gentlemen and women, they are doing exercise, they are running, they are working, and their lifespan is very much high than the person belongs to India. I think it's almost their lifespan is 80, while in India it's 60, and some again it's declining. So exercise is very much necessary to reduce the cancer risk as well as to make you fit. So obesity, yeah, obesity is another risk factor. So what is that actually? So when your weight will be higher than the healthy weight, then you will be obese. So we can say that's obese. So what should be the healthy weight? So as we can say that we everyone know that BMI, how to calculate its weight in kg, then it's height square divided by height square. So if you see that, yeah, if it is below 18.4, then you are underweight. Then if it is normal actually from 18 to 24, but if it is more than 25 to 39, then you are overweight, and if it is more than 40, then you are obviously obese. So yeah, body mass, that's the same thing which I have spoken, it is written here. So if we see that the obese people are at a risk group for 13 different type of cancer. So we can say that. So thyroid, breast, liver, gallbladder, upper stomach, pancreas, colon and rectum, and following by like kidney, uterus, ovaries, esophagus. So obesity is obviously a main, a very much significant risk factor for developing cancer. Not only one, for it's relating with different type of cancer. So we should maintain our weight. So why it is like overweight and obesity can cause changes in the body, including like inflammation. It can increase inflammation. So how higher than it can have the insulin higher than the normal level, and also some sex hormones. So these changes are the responsible to develop cancer in future. So we should not be obese. We should do exercise and we should lose our weight. So these are the which I have told up to this time. These are like our demographic factors, lifestyle habits. But now like pathogens. So in the pathogens which are related to cancer, it's like bacteria and virus. So coming to bacteria, so if you see the bacteria, the most dangerous bacteria which is associated with cancer, it is Helicobacter pylori. Helicobacter pylori is also it's an infection. So it is responsible for the stomach cancer. So most of the it can means, and the men are having more susceptible to get gastric cancer than women due to this Helicobacter pylori also. So Helicobacter pylori is having a virulence factor which called Kg, which everyone may have this Helicobacter pylori, but all the strains are not dangerous. All the strains are not responsible for developing cancer. But the strains which are Kg positive, they are more dangerous to develop the cancers. So like, and it can be transmitted by saliva. Even it can be transmitted by fecal contamination of food or water. And then in the developing countries, a combination of untreated water, which we used to have sometimes, that it can also, if it is poor hygiene, it can also the responsible for this bacterial infection, and in future, which is going to give you a cancer in stomach. So virus, if you see the virus, so many viruses are there which are related to cancer, like Epstein-Barr virus. Actually, it is a virus which is spreading through the saliva. And when I did my research in Mizoram, I got it that in Mizoram, Epstein-Barr virus is very high compared to other parts of India, even other parts of Asia. So this is maybe a very, 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 very dangerous factor for developing gastric cancer. Basically, what it used to do, in human body, it used to do the methylation, and then it's going to be dangerous for developing cancer. So coming to hepatitis B, this is another virus which is responsible for liver cancer. So again, human papillomavirus, which is a very dangerous virus also. We can tell that it's having 12 strains, and it's mainly responsible for cervical cancer and for other cancers also. So these are the things. So coming now, I want to tell why individuals itself is a risk factor. So like breast cancer, if your menstrual periods at an early stage, then it's a cause of risk for developing breast cancer. And then if you're later stage, then also it is. And having child at older age, then also it is a risk factors. So again, you have to know that family history or genetics of your family. Actually, in your family, maybe it's sometimes some persons are there which is having earlier gastric cancer or maybe breast cancer. Then you are in a risk group if your family before having any cancer, then because it will make your genetic mutations. So now it's genetic alterations. So polymorphism and mutation, it can be divided, like if it is more than 1%, then we can tell that gene in your DNA sequence, then we can tell that it's polymorphism. And if the changes in your DNA sequence is less than 1%, it can be a mutations. So mutations can be like substitutions. So that means maybe here, if it is, you can see 82, it's become C2G. So it's a substitution. It's substitution mutation. Coming to insertion, so here I can see that one more bases has added compared to wild type. So it's like insertion. And coming to deletion, it means one base already gone. So these are the different types of mutation you can have. So here you can have repeats also. You can see that CG and AT, it's continuously coming. So these are kind of mutations which can cause harmful mutations in later on, which are responsible for developing cancer. So in case of gene, maybe it's copy number, it can increase, it can duplicate, or it can deletion of your particular gene, which is responsible also. So if we see the mutation, it's like synonymous and non-synonymous mutation. So if you see that HAT, I give an example only. So if your A is going to change like a small a, so the meaning is still same. It's a synonymous mutation. It's not dangerous. But if it is going to be HUT, it's your mutation is going to be dangerous. So then you have to calculate that if it is a mutation, then it's natural or benign. It is natural or pathogenic. So these are the tools. I'm not going to details here. These are the tools by which you can say your mutation is pathogenic or not. And this effect of mutation can be its gain of function, loss of function, and then it can be spontaneous, it can be germline, it can be somatic. So we can have so many type of genetic alterations. So like BRCA1 and BRCA2 mutations for breast cancer, it's a signature. If you have a BRCA and BRCA2 mutations, you can have a risk of getting breast cancer in future. So at last, I want to say that, between 30 to 50% of cancer can currently be prevented by avoiding risk factors and implementing existing evidence-based prevention strategies. And again, the cancer burden can also be reduced through early detection of cancer, which maybe my your next speaker is going to tell about the early detection and treatment. And then many cancer have a high chance to cure if diagnosed at early and treated opportunity. So at last I want to say that, yeah, there is a can in cancer. So surely we can overcome this problem one day. Thank you for listening this talk. I have finished my lecture.
Thank you so much, madam, for this wonderful session, wonderful explanation. And with this, I'd like to call upon Dr. Rumki Nathan, HOD, Department of Zoology, Holy Cross College, Agartala, just to introduce our second resource person, Dr. Shovik Ghatak sir. Over to madam.
Good afternoon. Good afternoon, everyone. Good afternoon to Father Benike John, Principal, Holy Cross College. Good afternoon to the respected resource person, the faculty members of Holy Cross College, and my dear students. We have amongst us today a very eminent resource person, Dr. Shovik Ghatak. We welcome you, sir, today to one-day international level cancer awareness workshop. Sir Shovik Ghatak has done his BSc from Burdwan University, West Bengal, his MSc from Berhampur University, and PhD from Mizoram University. And sir's dissertation topic was analysis of mutation in key trigger genes associated with gastric cancer from subtyping to early prognostic assessment. Sir has got many prizes and distinctions. Few that adds to his credits includes Ramalinga Swami Re-Entry Fellowship from Department of Biotechnology, DBT India, RSBO Fellowship from Canadian Society of Bioinformatics, Young Investigator Award from American Gastroenterological Association, USA. He has also been awarded as the best oral presenter in Second Indian Cancer Genetics Conference and Workshop, ACTREC and Tata Memorial Hospital, Mumbai, India. He has also been awarded with the best paper poster presenter in Advances in Cancer Genomics Conference, Mizoram University, Aizawl, Mizoram, India. Sir has received many fellowships and grants, which too adds to his credit. And amongst these are research grant, which he got thrice, one after another, subsequently in the year 2021, 22, and 2023 from the Royal Physiographic Society of Lund, Sweden. Sir has also received bioinformatic grants from RSBO Fellowship Competition, Canada in 2021, 2022 to 2021. He has also received DBT Research Associate Fellowship and Senior Research Fellowship from DBT State Biotech Hub, Mizoram University, India. Sir is presently working as Senior Associate Scientist in Lund University, Malmö, Sweden. And his current research focuses on prostaglandin D2 and its receptor signaling, enhanced cancer cell stemness, and tumor burden in human colon cancer using varied cellular, biochemical, genetic, molecular approaches in cancer patient samples, cancer cell line, and model organism. This is not just the one that adds to its credit. Apart from his present association, Sir has also been associated with Mizoram University as Research Associate, as Senior Research Fellow, as Technical Assistant for quite a long duration. Sir has also experience of postdoctoral fellowship from Beckham University Institute of City of Hope, Monrovia, USA, and Simmons Cancer Center, Bangalore, Scott and White Health Research Institute, Dallas, Texas, USA. Sir has got 28 papers published in renowned national and international journals. So he is an eminent researcher in the field of cancer technology. And with these few words of introduction, I once again welcome you, sir, Sir Shobik Ghatak. Sir, I believe and we all believe that your session will immensely enrich our budding minds. The platform is all yours, sir.
Thank you, ma'am. It's very nice introduction, but it's too elaborated. So I can start my session, and before that, actually, I'm going to talk mostly about how to detect the cancers and how to cure the cancer in a basic scenario wise. Uh, just a story I will tell about it. So I can share my screen first. It is visible now.
Yes, sir,
Yes,
it
sir,
is
it's
Yeah,
visible.
visual.
yeah,
Yes.
yeah. So today I'm going to talk about just how to detect the cancer in early stage or in pre-malignant lesions, like before starting the cancer, and as well as how to prolong the survival for these patients. So now you can see some lighting here going on. Just it's a black and red lighting, and some people are standing there. So this is kind of a detection. We don't know that where is our tumor or where is our cancer's particular sites. So we can't know from outside. So we need to identify that thing. For that, we need to do scan our body. Mostly the early detection is possible nowadays based on the like result of the CT scan or otherwise MRI. These things is available in our hands, but it's very difficult to identify in CT scans or in MRI that tumor is where it is present. If it is a early stage, means if it is a very small tumor, so that time it is detection is very difficult. So we need to develop some technique or some kind of a molecules or some kind of markers which can identify that our body is uh, taking a particular tumor, and that is a pre-malignant or otherwise it's a early stage like stage 1 and stage 2. Dr. Chakraborty already explained that about the different tumor staging, so I am not going to come in the tumor staging part, but from that, for starting early detections, we need to know about little bit about the cancer, how it is developing in our body. So what is cancer first? It was elaborately explained by Chakraborty, Dr. Chakraborty, but I can just show that a uncontrolled cell growth is a cancer. Means every cell is growing in our body, but not every time it is maintaining the same time. Uncontrolled cell death means if it is developing faster or it is developing in multiple number than the usual number or usual times. So that will be the tumor growth, and that tumor can be formed a malignant, and that will be the cancer. And another thing is if your cancer is developed in the early stage, like stage 1 and stage 2, it will be the uncontrolled cell death, cell growth. But when the cells is started develop like growing, that time it can come through your blood vessels and it can migrate through your blood vessel to another secondary sites, and there it can start growing again. That will be your stage 3 and stage 4 cancer. So that will be your highly risk condition. So two type of different condition we have. One is early stage, one is stage 1 and stage 2. That will be the just uncontrolled cell growth. And if it is migrating towards in a secondary sites, that will be your stage 3 and stage 4. That will be the late stage. So these will be mostly important. So detecting a cancer in early stage is the best condition. If your cell already started migrating, that will be difficult to track back, and as well as that will be very difficult to control. So better to develop a marker which can identify in stage 1 and stage 2 condition or otherwise in before pre-malignant condition. So with this, if you see from normal cell to cancer cell, how it is developing the cancer from normal to cancer cell. In that, in your normal cell, you can see that epithelial cells are there. Hmm? Yeah, normal cells. It is visible now. Hello.
Sir, screen is visible, but slides are not visible.
Slides are not okay. Okay. Sorry. Sorry. One minute. Now it is visible now. The slides. Yeah.
Yes,
I
sir, it
was
is visible.
explaining for this one. I was explaining for this one. I'm not repeating, but you can understand from my talk that we can scan our full body and we can detect our tumor. And then the uncontrolled cell growth is the stage 1, stage 2, and foreign cell and interacting cells when it is migrating from one part to secondary part, like primary part to secondary part, that will be the stage 3 and stage 4. So with this, if you see how the cancer is developing, like tumor is developing, we have normal cell in our body everywhere, mostly the epithelial cell at the mucus level and the endothelial cell at the upper level. So when the cancer is trying to form, like if you get any mutation or if you get any exposure, the cells is going to change their structure. Mostly first the epithelial cell will change, then it will come to the dysplasia. That time epithelial and endothelial cells both will change, and then after that the cancer will form. When the tumor will be bulging out or it will be more bigger size, it can penetrate the blood vessels and it can enter inside the blood vessel. That will be really a like a cancer condition or malignant conditions. Then everyone, like sometimes we used to get that in our from our skin some bulging tumor is coming out, but all the tumor are harmful or not, we need to know that thing. There is a two different kind of tumor. One is benign tumor, another is malignant tumor. Benign tumor are stuck on the particular organ and it is non-cancerous and it can migrate through the blood vessel to the secondary sites, and it will not grow faster like a malignant tumor. So benign tumor are not that much harmful, but if you are getting a benign tumor or adenoma, better to go and take some medicines or otherwise you can operate that one. Because benign tumor can form a malignant tumor in later stage if you are having a different kind of exposure. So what is malignant tumor? Malignant tumor are the cancerous most. If all of you know that TP53 mutation is the major factor for the cancer formation. Most of the cancer has lethal TP53 mutation, which is controlling our cell cycle. If TP53 is mutated, then the cells will have uncontrolled growth and it can migrate through blood vessel. That will be the malignant tumor. And in this condition, you need to identify it in an early stage and as well as you need to cure this particular tumor, malignant tumor, as early as possible. So in next, we used to talk about the cancer, but cancer is not a like a single thing. It's having a multiple number of different variations there. Mainly, it has 4 different variations. First is the carcinoma, that is the most common, because if you are getting the cancer in the colon, stomach, oral, breast, all are in the epithelial cells. That will be the carcinomas. Then the sarcomas. Sarcomas is very lethal cancer than the carcinomas. It starts in the cell in the supporting tissues of the body, such as bone, cartilage, fat, connective tissue, and muscles. So if it is happening in your bone, it will be very difficult to cure. So sarcoma, that's why more lethal. Then lymphomas. Lymphomas are the main cancer part which is happening in our immune system, mostly in T cells or otherwise in different B cells. So these particular lymphomas can be cured by using some drug only, not by surgery. So this is more lethal. Then leukemias. Leukemias are the major cancer related risk for the child mostly, and sometimes in the middle aged people, because for this particular cancer is to form in the bone marrow. If you can't transplant your bone marrow, this cancer can't be cured. So that's why it is more lethal, leukemias. So these major four cancer types are available, and others also cancer types are there, like adenocarcinoma. Inside this carcinoma we have different different type of cancer, like adenocarcinoma, like mucinous carcinoma, like syncytial ring cell carcinoma, all these things. But major all these four cancers are available in our clinic. So first I'll come one by one, the carcinoma, sarcoma, lymphomas, and leukemias. How we can develop some different kind of marker for this. So all these four different type of cancer caused by two different things. One is invasion, another is the metastasis. Invasion is the early stage and metastasis will be the late stage. In invasions, the cells, if you see this particular animation, here you can find it out that cells from the primary tumor, it will invade to the blood vessels, and then it will migrate to, and after that it will create a new secondary tumor in another particular organ. That will be your metastasis. And if you have the metastasis, you can't know that where it is happening. If you have a stomach cancer, the metastasis can happen in lung or liver or brain. If you have a breast cancer, your metastasis can happen in the brain or lung. If in that scenario, metastasis is to occur. But in the clinic, you can only treat a particular organ, not all the organ. If you treat more patient with a different drug combination, so you will get more toxicity and you will get more side effect. So patient will die very early. So that's why we need to think that first we need to identify this cancer in early stage. So the cancer cell will not able to migrate and it cannot make the metastasis or secondary tumor, which will be very tough to cure the patients. So that's why it's important to identify a marker which can detect in early stage. In next, if you see the statistics in global cancer statistic in 2022, mostly estimated new cases are near about 1.4 million in men and 1.3 million in women. But in men, the highest is the prostate and the lung and stomach and colon and rectum. And in women, obviously it will be the breast. But if you see the estimated death is huge. Like for men, it is 49,000, more than 49,000 men is dying in every year. And for women is 31,000, more than 31,000 women are dying. For men, it is like lung cancer. For women, it's also lung cancer. Because this is only for the habit, dietary habit as well as your lifestyle habits. Lung cancer used to happen mostly for the smoking habits. If you are regular smoker, then the chances will be more than 80% for lung cancer in your older age. So that's why smoking is the major factor for the like cancer related death than the cancer related incidence. But most important is the death for us. So that's why smoking need to stop, otherwise people will die very early. Then I'll come to some like history about the cancers, because for developing a marker you need to know a little bit about the history and why we want to go for the marker research, early detection marker research. For that, if you see, like 1970, the survival rate was very less. Like from 4 people, 1 people will survive only 1970. But after that, the different researchers are trying to identify different early detection marker or different treatment strategy. So they have improved little bit. Like from 4%, 2% are going to be survive after treatment or after detection. But if you see the next 20 years, like up to 2033, like that, or 10 years or 20 years, you can see the research are going on in a such a way that we can identify some early detection marker which can be detect these particular patient in pre-malignant lesion, like before formation of the cancer, as well as it can detect in the stage 1 and stage 2 patients only. So we can easily cure that patient after surgery or using some different kind of drug. So important thing is as much as possible research, if it is going on to identify the early detection marker, that will be more important for different geographical location. If you see the statistics wise, mostly in 2013 up to 2013, the number was increasing too much. But after that, for male and female, it is reducing. Why it is happening? Because for the all the research is going on to establish a early detection marker as well as different drug combination, which can be useful to prolong the survival of this patient. So that's why our cancer research group is every time growing. It is not declining. But in Indian scenario, it is little difficult because in Indian scenario, lots of people are from different ethnic community, as well as they are having a different kind of food habit, different kind of lifestyle habit. So controlling in Indian scenario is bit difficult. So in India, it is not like this particular case. It is increasing still now. Up to 2010 it was declined little bit, but after 2010, like 2015, it started increasing again. Mostly in the northeastern part, if you see, it will be the hotspot for cancer, different kind of cancer. More than 15 different type of cancers are the hotspot for this northeastern Indian part, because for the lifestyle habit. So we need more researchers from this particular region because we know our own culture from the northeast, and we can treat our patient in that way. Because if we have knowledge, you can treat that patient. If one person is coming from outside, they will not able to know their culture, their habits, their quality of life, their socio-economic status, all these things. It's impossible to learn. So, but if you are there, so you can go for more cancer research, and you can develop some marker or develop some drug which will be useful for these kind of regional patients. For these things, after these things, we got the statistic. But how to prevent this cancer? It's difficult to prevent the cancer without this factor. If we can stop smoking, major of the cancer will be reduced. Then keep your healthy weight. Like you need to maintain your BMI as well as your weight and height. Eat as much as possible green fruit, green veg, every day, like vegetables. Then drink alcohol less. That will be very much effective for different kind of cancer. Then this sunlight or UV light will be not that much problematic for India, but in Western country it is too much problematic. And eat less processed or red meat. Like if you are eating too much pork meat or if you are eating too much goat meat, it will be very much dangerous for your health. It can increase your obesity, and you can be obese. Then it will create more different kind of hormonal changes in your body, and it will reduce your immunity. So you should eat less more red meat or processed meat. Then mostly fiber diets are very good for a different kind of diseases. Then activity and exercise, and more of main thing is the eat less salt. Salt is the major factor for your ulcer or for your gastric cancer, for your colitis. All these things used to happen after eating your more salt. And as much as less raw salt if you are taking, that will be better for your health. But other factors also are there. They are mostly you should not exposed to some harmful virus or bacteria like HPV, H. pylori, EBV, HIV, hepatitis B and C. Then for your work purpose, like if you are working for mining and all things, that will be also a risk factor because you are exposing for more asbestos and as well as different radiation and all these things will be major factor. Try to reduce these things, then we can easily control the cancer and we can prevent. But although these all things are in our lifestyle as well as in our daily routine, so it will be very difficult to control everything. But we try to control these things, then we'll reduce our cancer. Now I'll come for the early and late cancer diagnosis. Early cancer and late cancer is the same thing like I explained before. Early is the stage 1, stage 2, late is the stage 3 and stage 4. But if you see in early cancer, if you diagnose the patients, like in stage 1, stage 2, 54% patient will be survived more. If it is in late cancer, 46%. So near about 10% difference is there between these three. So that's why we need to develop the early diagnosis marker. But it is not common for all cancer. Different cancer are having different statistics in this scenario. Like for breast cancer, if you are in early detection is happened like BRCA1 or BRCA2 mutation study, if you do in early stage, then you can survive, you can make 85% more patient can survive. Then bowel cancer, it is near about half and half 50%. But lung cancer, it's not like that. 75% patients will die in lung cancer if it is the late. And melanoma and skin cancer is easily curable in these days because there is a different kind of immunotherapy, different kind of drug is there. So if you detect in early stage, it can increase 91%. Then ovarian cancer also, it is 50-50. So we need to have more research in bowel cancer, ovarian cancer, lung cancer. We need more people to do this research because there is no certain early detection marker are available in this type of cancer. Now, if you see the survive by stage at diagnosis, why we need the early detection marker? Because if you diagnose it in earlier stage, from around 10 patients, 8 patients will survive in longer period, like more than 5 years, it can survive. If you diagnose in the late stage, only 2% will be survived from 10 patients. So it is very difficult to identify the lung cancer in early stage because you can't get the symptoms here. We need some marker or we need some molecules which can detect that in our lung some inflammation is there, or it is in a pre-malignant stage, or it is in a just tumor formation is started. So that's why lung cancer researchers are more required in Indian scenario. If you see the bowel cancer is little bit different. If it is in late stage, detection is there. Among 10 patients, 4 patients can survive up to 5 years. So bowel cancer is also important. But if you see the colon cancer, in late stage, if you detect, then more than 5% will be survived if it is late stage. So why it is happening? Because in Western countries, these particular cancers are not prevalent before. The colon cancer was more prevalent in Western countries. So they started doing lots of research and developing early detection marker. And they developed some early detection marker which can detect in early stage for colon cancer. So the research was more advanced for colon cancer, but not for other cancer. But nowadays, lung cancer are more prevalent. So Western countries are going for the lung cancer early detection marker development. In India, lung cancer also is major factor because for this smoking habit, most of the like college students or university students or older age peoples are going for the smoking. And the quality of the smokings, like they are using some locally made some smoking things or unpurified tobacco they are using for smoking. All these things is the major cause for this lung cancer in Indian scenario. For coming to this particular like breast cancer, I'm talking about all different kind of cancer. So how we can use the early detection for different type of cancer. So if you see the breast cancer, it is happening in throughout the world. in every ethnic community are suffering for this breast cancer for female, but most of the young females are suffering for that. So we need to develop a marker which will be able to identify it in the young people, because in the old people is getting breast cancer is very few, but the young peoples are getting more. Why this is happening? Due to our lifestyle habit, because young people are fond for these particular smoking habits or different hormonal therapy, different kind of lifestyle habit, so they are getting too much. But if the young people are dying in a young age, so it will be difficult for our country to maintain these particular status, because young people are the most responsible to develop our country. So that's why we need a early detection marker which can identify the young people breast cancer or other different type of cancer, and which can be translated to the clinic. So for this, breast cancer research is also very much important. The early detection marker for the breast cancer is very much important. Next, if you see that why and how we are found for this diagnostic marker or early detection markers, how it came. If you see the human genomics evolutions, first, the Mendel proposed some theory, but that time we don't know about this much in the cancer or different biomolecules. But throughout 50 years, we developed lots of techniques, lots of different platforms, lots of different biomolecules which can detect the cancer based on the human genome sequencing. 1999, we got that first human chromosomes. After that, it's just boomed the research research for this human genomics. And when the human genome project came in 1990 and it's completed in 2003, the first genome cost was 2.7 billion in total. It was very difficult for the people to do because 2.7 billion dollar is huge amount. So that time people doesn't think that we can have our own diagnostic marker or personalized diagnostic marker, but it's reduced day by day. Now it can possible in a $1 for human genome these days in American scenario. But in our like developing country in India, we have like limited resource. So we can do up to like 10,000 rupees. We can do our whole genome in the next generation sequencing. Then how these particular early diagnostic marker or personalized medicines came. One paper came in the science that human genome total sequence, but we don't know what is happening inside the human genome. That time they discovered that which genes are responsible for different type of diseases and which genes are responsible for these cancers. That time we identified different genes like T53, different kinase genes, different like growth factor genes, which are responsible for the tumor growth. So from that time, we can identify that which genes should be targeted or which genes should be captured for early diagnosis. So this is the different era of different genomic study as well as the progression of our clinical like improvement in throughout globally. Now I'll come for the personalized medicines. Personalized medicines means we can create our own medicines for a individual person. This came in 2015 when American governments they have started a particular society which will identify from this human genome project, because human genome project is after the thousand genome project came. In thousand genome project throughout from Europe, America, China, in other parts like from India, some Gujarati people also there, and they have sequenced whole genome, and then they identified which particular genes is responsible for their disease and which particular genes is abnormal. So we can target that genes to cure different genetic disease like cancer. So that will be the personalized medicines. And for these personalized medicines, when it came, people thought that it is easily curable for the different cancer or genetic disease. But some challenges we face still now. like delays in diagnosis, like in Western countries, UK, like in Sweden or America, we have a kind of a like pipeline that every people should have their own checking for that breast cancer or like gastric cancer or like colorectal cancer screening. So they are having already different screening procedure, like they are using some different kits or different scanning method to identify this particular person is going to be have cancer or not. That is in a pipeline. But Indian condition, that is not possible because people mostly afraid. They never used to go to the clinic, and that's why the diagnostic will be in very delayed. And when the person is getting stage 2 or a stage 3 or stage 4 tumor, that time people are diagnosis, and so that time the chances for the survival will be very less. Then the second point, limited robust evidence that treating patient based on the molecularly predicted primary improve outcome. Like for this means we have very less evidence because the research are very limited in Indian condition or underdeveloped or developing countries. So we can't get so many evidence that particular genes is responsible for that cancer. So that's why more research is required to identify these diagnostic challenges and overcome these diagnostic challenges. Then several research and commercial tissue of origin classifiers means if you know really about the tumor. Tumor is not a same characteristic cells are there in the tumor. Different cells having different phenotypic characteristic, different genotypic characteristic. So that's why it is very difficult to identify a particular tumor that which genes to be target. So that's why we need a recommended diagnosis which is not available in our clinic. Then we don't know the tissue origin. If you have the secondary tumor, we don't know the tissue origin, the primary tumor where it is happening. Because this, in the secondary tumor, whatever the tissue is there, that's coming from the primary tumor. So the characteristic of the tumor is the like primary tumor. So we need to treat that secondary tumor like the primary tumor treatment. So that's why we need to identify the origin of this primary tumor. So we can develop a novel therapeutics or immunotherapy based on the primary tumor origin. Then the profiling of the tumor tissue are mostly molecular profiling are failure at 10 to 20%. Means although we have developed lots of molecular techniques, but still we have a limitation. We can't check throughout all the cells in tumor because different cells is having a different kind of mutation. Different cells is having different gene expression. So molecular profiling is failure day by day, and researchers are developing new techniques like single cell sequencing they have developed. So from this tumor they will target it or like do the sequencing for each particular genes for each cells to identify what type of tumor phenotypic and genotypic profiling. So that's why it is improving, but still we failure to identify all these different type of cells in a single tumor. And in clinic, mostly we used to take a small biopsies, like if you have a gastric cancer or if you have a colon cancer. So if you have gastric cancer, we will go for the endoscopy. And that time, endoscopic will take just a pinch of a tissue. And that tissue will not able to reflect total tumor. So that these things we need to change little bit way. But how to do it? It will be very difficult. Patient will be in a like tough conditions because it will be too much painful for them. If you take a bunch of tissue from the like stomach or bunch of tissue from the colon, so it will be very difficult to take huge amount of tissue from the patients because they are already in pain. So to overcome these things, we need to develop a such kind of molecular marker or early detection marker which can be easily available. So the alternative is liquid biopsy. Liquid biopsy means is just in our body fluid. I'll explain why we need these things. Suppose from this example we can see that we have a particular tumor in the stomach, but we don't know that tumor is how much growth is there and how much molecular characteristic is there and all these things. So if we collect the liquid sample like peripheral blood we can easily collect, stomach wash or gastric juice we can easily collect, and we can collect in a huge amount, and urine we can collect, saliva we can collect easily. So if you use these particular fluid for making a biomarker which can use for early diagnosis, that will be useful, because we need a particular molecule which will be more sensitive and as well as more specific. Specific means particular molecule which is available in the blood, saliva or urine. It will be just reflect the stomach conditions. The tumor is present in the stomach, so that's why we need that same condition biomolecules. And as well as sensitive means it should be able to identify it in the body fluid. Tissue is the best things every time for any diagnosis or anything, but taking tissue is difficult, so that's why we need body fluid. So in the body fluid, what are the things is there? Different types of biomolecules, because all of you know that in our blood or saliva or urine, there is the mRNA, there is a different circular RNA, there is a different tRNA, different protein, different vesicles is there. So we can collect all these vesicle, mRNA, DNA, and tRNA, all these things, and we can go for their profiling. How we can do the profiling? We can just sequence mRNA, we can just sequence microRNA, we can check their expressions, that how much mRNA is producing in the particular body fluid, how much microRNA is producing in the particular body fluid. Then cell-free DNA, the DNA is coming from the tumor, that particular DNA is having how much number of mutation and how much number of copies coming from this tumor, that will be the cell-free DNA. Then CTCS. CTCS means the circulating tumor cells. Because when the metastasis is happening, that time the tumor cells will migrate through the blood vessel. If we collect the blood, that time we can capture that particular CTCs and we can check their profiling. So we can identify the primary tumor origin, from which part of the body this particular circulating tumor is coming. And the different secreted protein, like if you know that different tumor markers are available in clinic, like CA, then CA15, all these tumor marker are available. These are the secreted protein. If you have inflammation in your body, so that particular protein will be secreted in your blood or like in serum. So you can detect that protein, secreted protein, and we can check the level, how much it is increasing or it is decreasing. If it is increasing, so it will be the risk for the cancer. So like that, we can identify early diagnostic biomarker from different body fluid samples. Why we are showing this body fluid? Because if you want to identify the early diagnostic marker, it should be in pre-malignant lesions, pre-malignant stage, or otherwise in your early stage, like stage 1 and stage 2. In your pre-malignant stage means that time tumor development is not there. So people will not go to the clinic for diagnosis. So we will not get any tissue samples. We need just a serum sample that time, because we can collect blood sample from them and we can extract different biomolecules and we can identify their like amount, and we can say this person is going to be rich for this particular cancer in pre-malignant stage. And this called the liquid biopsy. Normal biopsy, what pathologists used to do, they used to collect the tissue, they will go for the pathologist test like histopathology and all, they will identify the tumor cells, that will be your normal biopsy. Now if we use this liquid like body fluid which can reflect that particular tissue, that will be our liquid biopsy. So in liquid biopsy, you can use in different phase of clinical application, like for early diagnosis and prognosis, like prognosis means we can identify this patient, how much survival for this patient, or how much time they are going to survive, or why these people died early. Then this is minimal residual disease means whenever you will have a particular like disease condition is a like a small tumor, if you are getting that time, you will not able to detect by the scanning methods. So that time we need something biomolecule which can detect the small tumor. So that can be possible. Then therapy selection means every patient will not have the same kind of tumor condition or tumor phenotypic or genotypic condition. So all the patients will not we should not treat with a same kind of drug or chemotherapy drug, or all the patients should not be selected for the radiotherapy. So that need to be identified. And if you are treating all the patients with the same kind of chemotherapy regimen, that patients will go for more toxicity. And these patients will go like survive less. So that's why we need to identify the patients based on this liquid biopsy, that which patient is going to get this which kind of therapy. That is also a response to the therapy also, because when you will treat a patient with a drug, after that, this patient is cured, but a secondary tumor will form again. So you need to identify it, that why this secondary tumor is again is coming, because already we have done the surgery, we have already treated the patients with a different chemotherapy drug, but why it is happening? So that's why we need to identify the resistance of these cells. In tumor, if some cells are resistant for this particular chemotherapy drug, that cell will not be killing by this drug, or that cell migrates to from primary to the primary tumor place to the secondary place, and that can form a bigger tumor. That will be the relapse. So resistant cell identification is also a preliminary things for the diagnosis. Otherwise, we will not able to treat the patient. So using this liquid biopsy, you can do all this clinical application. How we can do it? Like if you take the blood, plasma or serum, you will have the CTC, because earlier I have described that circulating tumor cells, circulating microRNAs, exosomes, then TAPES is the tumor educated tumor educated uh, your platelets. That platelets will go through the cell like the blood vessel to inside the tumor, and they will create their own characteristics, and that can be also identified, and that can be used as a diagnostic marker. Then the circulating tumor DNA, to identify it in the body fluid, that what are the DNAs there, how their mutation condition, how their copy number condition, how their methylation condition, and we can identify that patient might going to get the cancer. These all things we can capture from the plasma, serum, or otherwise saliva as well as urine. And in this CTC, we can use for further research. Like we can derive organoids, means this CTC is coming from the tumor, so we can culture in our lab and we can identify that which drug can kill these particular tumor cells. That we can do in vitro or in vivo, different mice model or different zebrafish model using this CTC. So this is very much important liquid biopsy. Then other things, what we can do, like chromosomal aberration study, methylation pattern study, amplification, deletion, and point mutation, translocation, different expression, like RNA expression and protein expression. And we can build a cutoff. From that cutoff, we can identify which patients are in high risk and which patients are in the low risk. So these are the all different available liquid biopsy application is there in research, like in cancer research. So I'll just explain a brief procedure how to do the early diagnostic marker, because it's not a like a 1 year or 2 years work. It needs more and more research, like it needs more 10 years, 12 years research, then only you can develop a marker. So the first things, if you have the like a cancer in your body. You can collect the blood, plasma, or serum, and you can extract the biomolecule like different kind of RNAs, like cell-free RNAs, exosomal RNAs, and microRNAs, tumor DNAs, different cell-free DNAs. That one need to be sequenced and identified their expression pattern. Because we are going to sequence, we are not going to using like normal Sanger sequencing or gene by gene sequencing or otherwise real-time PCR to identify the expression of a particular genes. We are not doing because we know that cancer is not a single factor disease or single gene mutated disease. Every cancer, every tumor has having lots of mutation, lots of genes are abnormally expressed, lots of genes are abnormally methylated. So we need to identify the bunch of genes which we can study in later stage. That's why we are going for a bulk sequencing. And from that sequencing, we'll use some bioinformatics analysis or clinical statistical analysis to identify the best possible marker, which can be captured from that sequencing and which can be reflect for that disease condition. And we'll go for the gold standard method like a real-time PCR or Sanger sequencing for a large-scale validation. If you sequence for 50% for sequencing, then we need to do the validation for 1,000 number of people, 1,000 number of patients. Then only we can easily and strongly claim this particular biomolecule can be used for early diagnostic marker. Then after that, we need to identify a particular cutoff. Cutoff means if you want to bring to the clinic or if you want to bring this marker for the screening in the large scale population, you need a particular number or a particular like amount. That amount will reflect your normal condition, and above that normal condition will be the high risk patients. That needs to be done using some statistic and bioinformatics analysis, and we can identify a specific cutoff, and we can tell the clinician that if it is more than this cutoff, the patient is in the high risk condition. And we'll identify these patients like high risk and low risk. In this example, if the selected RNase is down-regulated, the patient is the low risk. The green color human body are the low risk, and the red are the high risk because this particular gene expression is upregulated. So like this, we are identified a early diagnostic marker. It's just a brief example that how to develop a early diagnostic marker for clinic. And but this research is not like a 1 year or 2 years research. It needs more and more times. And after these things, we need to go for the clinical trial in a mass screening in a different geographical populations. So with these things, first we identified the patients, we identified this high risk group of patients, then what we will do with that patients. With that patients, we can treat the patient in different conditions. Like if it is a low risk, we'll treat with the normal drug or normal like procedure we'll follow in the clinic. If it is a high risk, we need a specific kind of medicine for that patients, which will be only for that patients. That will be the personalized medicine for that patients. And for that personalized medicines, we need 5 rights, and that 5 rights need to be followed in the research. One is the right dose of this particular medicine. That we can identify it from the genomic sequencing or pharmacogenomics of that patient. Then the right drug for that patient, because all the drug will not fit for that patient because their tumor genetic characteristics is different. The gene mutations are different, gene expression are different. So we need a particular drug which can target that gene only. So we need right drug for that patient. Then the right indication for means if you treat that patient, we need to identify that patient is going to be benefited or not. They are responding to that particular therapy or not. So that's why we need a indication that patient is are like they are going to you like cure using this drug. So that thing also based on the different biomarker we can identify. Then the right patients at means all the patients we will not able to treat in the same condition because patients age will be differ. Patients immunological factors will be differ. Patient immunological burden will be differ. Patient gender will be different. So that's why we need to identify it that which particular immunological cells we can target with that drug because all the patients we can't treat in the same conditions because some patients is having chronic disease like thyroid or cardiovascular disease. We can't use the same drug although they are having the same mutation. Then also we can't treat that patient in that same using their same drug due to their condition. Then the right time for means we can't treat the patient in longer. Like if you treat these patients with a aggressive drug, that patients might suffer with toxicity for longer duration. So that's why we need to identify the right time to identify that particular patients will benefit up to 6 months. We'll treat the patients up to 6 months only, and that can be possible after developing a marker for all these thyroids. So these are the different personalized medicine strategy as well as we can develop the marker for early diagnosis as well as for this particular treatment. With these things, I'll conclude it because I'll not take so much time. And thank you for these things to give me these opportunities. Thank you.
Thank you, sir.
Thank you, sir. Thank you, sir, for the wonderful deliberation. I hope we all are immensely benefited, especially our students. I also thank our first resource person, Dr. Payal Chakravarti. And now the topic is open for discussion. Both the topics are open for discussion. Students and faculty members, any queries, any question, both the resource person are available. So you can use the chat box and also you can ask the question by unmuting the microphone. Sir, ma'am, in the meantime, our students, they get ready with our question. Can I ask a
Yeah, yeah,
question?
yeah,
Like,
yeah, yeah.
sir, cancer at different stages of development, like requires different approaches of treatment. Like, what would be your suggestion, sir? Like, if it is diagnosed at a very early stage, whether alternative medicine can be opted as one of the way outs for the treatment?
Yes, that's why these days drug repurposing is going on. The research is going on in that way. So if you are able to like diagnose in the early stage, so you can't treat that patient with the same drug. You can take that whatever the drug is available in the market for other disease, and you can choose that drug is going to benefit it for the patients or not. And as well as you can treat the patient using some different generic medicines like the patient is suffering for the like different kind of genetical disease like in the cancer or diabetes or any cardiovascular disease. So you can choose some other medicine to boost their immunity in a such a way that immunity will trigger to suppress that tumor because these days immunotherapy is like it's a like a huge topic. People are using a different immunotherapy drug which are using in different diseases. That drug is coming for this cancer also to boost the immunity for that patient, and that drug will be benefited for these patients.
Thank you, sir. Thank you.
Yeah.
Any other question from the student side? Okay. Now I
Yeah.
will invite Dr. Sushmita Saha to deliver the both of slides for today's session. Before
Thank you,
that,
ma'am.
can I ask one queries to any one of the resource person? Sure, sure,
Yes.
ma'am.
Sure, ma'am.
Yes. Okay,
Sure, ma'am.
I'm Sangeeta Chakravarti, Assistant Professor, Holy Cross College. It's not a question, it's something like that. I want to know, like, to how to make it more, feasible enough, the early detection process. Because as you have mentioned that in India, it goes near around 10,000 to check on that one. If I'm not
Yes.
wrong on that, how to
Yes.
make it more easy for every common person to scan their body and to go for the early detection process.
It's throughout the research we can say in that way because it's like that if you are doing like if you spend that 1,000 rupees and you can do your whole genome or whole exome sequencing and you will identify that some mutation is there, but all the mutation is not responsible to develop the cancer. One thing. So that's why you need a clinician to identify which mutation is causing for this particular cancer. Like in breast cancer, ovarian cancer, it's well like established that BRCA1 if you do, then you can identify that if you have BRCA1 particular one or two mutation, you will get the breast cancer in later stage. Or your family if having that breast cancer, you can go for the BRCA1 study. But in other cancer, it's very difficult. So that's why we need some liquid biopsy biomarker which can be detected in normal pathology. Like if you are having the cancer or if you are having stomach pain, you can go for the like clinic and doctors told you need to go for the endoscopy and you can identify that you will have the ulcer. That will be also early detection because if you have the ulcer, you will be in the high risk group. So in that way, in India, we can develop slowly, but it needs more time and more research is required because whatever the marker is developing in European region, UK region, or American region, that might not work in the India because Indian genomics and genetics are different for different like populations. So that's why we need more research in Indian people. These days, the Human Genome Project have started for India. Two different genome projects is going on, and one project is already finished. It's called the Indi Genome Project, and the samples was collected from throughout all the different community in all over India. Another one is the Genome India. That Genome India is also collected the different tribal region population as well as different like uh, different parts, geographical locations, population like base data, all these things, and they are correlating with the patient data and they are correlating with the normal like healthy population data also, and they are identifying that what are the different risk strategies is there like risk factors is there in their genomics like in their genes or in their like different secreted molecules are there, whatever the secreted molecule is there. All these things is going on, but it's required time to develop a screening, to develop a screening throughout the India for different kind of cancer. But I think for breast cancer, it's established because people used to go for the mammography. Mammography is the standard procedure for breast cancer. But for other cancer, it's very difficult. And as well as the virus screening is also important throughout in India, like HPV, different genotype screening, which is already developed in Western countries because every alternative 6 months you need to do the different virus screening from your saliva or urine or from the blood. So that kind of scenario need to be been in our like pathology or in clinic. Then only it can develop for the early detection marker.
Yeah, in this context, actually, I want to add that, yeah, as we both are trying to develop some marker, early detection marker for him, and for me, it's like a therapy based marker. So, if we can validate our markers in a greater number of patients, so in future, we can uh, try it in some clinics also to make it, means, is it possible early diagnosis of the patients. So let's see.
Best wishes and thank you very much. We are
Thank
very
you.
much hopeful that you all will come up with a brilliant ideas to contribute in Indian cancer history. Thank you.
Thank you.
Sir, I have a
Students are
question.
asking some questions. Yes, yes, yes.
Yes, sir, I am Dr. Suren Chakraborty. I am pursuing honors in image physiology from Holy Cross College. Sir, my question is,
Yes.
as the malignant property has been seen in the cancer, that is malignant tumor, can we go for the X-ray or gamma ray treatment?
Pardon me, I didn't hear you properly.
Sir, am I audible now?
Yeah, now audible. Yes.
Yes, sir. My question is, as the malignant property has been seen in the malignant tumor, can we go for the
Yes.
X-ray or gamma ray treatment?
It's not like that every time you can do it because most of the like if you have the malignant property in the tumor, you can't do every time the same chemotherapy or same same radiotherapy because it can affect to the normal cells, it can affect the surrounding cell, and which can create a new mutation, new methylation, and that will be effect in your normal life, and your survival will be not prolonged. Your survival will be reduced. So we need to identify the exact molecular characteristic of the tumor. Then only we can identify that which kind of particular therapy you can do.
Thank you, sir.
So one more question is there from Aishwarya
Yes.
Shinoy. Our question is there any way to dissolve benign tumor other than surgical procedure?
Yeah. It's there. Lots of drugs are available to dissolve the benign tumor, and it's based on the that stage because benign tumor is also having a different kind of stage. And if it is a fibroid adenoma or adenoma, they will be like better to surgery and they need to be taken out. Otherwise, it might be like malignant in later stage. If you have a early adenoma, you can dissolve it. There is some different kind of drugs is available which can dissolve that particular early adenoma. But if it is already fibroid adenoma or it's a late stage adenoma, better to do the surgery.
Thank you, sir. One more question is there. Is there any universal cancer marker for all type of cancer? This question is from
Uh,
Anna Lakshmi.
yeah. Yeah, there is not like that universal cancer marker because all the cancers are different and all the can tumor like cell stage are different. So it will be very difficult to identify a single marker for every kind of cancer. But better to like in the clinic in the Indian scenario, the practice is there. First, if you go to the clinic, they will do the tumor marker. Different established tumor marker is there like carcinomembranogenic antigen. If you have a normal inflammation in your body or tumor proliferation is there, so the carcinomembranogenic antigens will be secreted in your bloods and it can be detected. But the like sensitivity and specificity is not that much good. So if you able to detect by this a single marker, it will not be reflect that you are having a high risk condition or not. So that's why required more research and identify different particular molecules which can detect different cancer.
And Hasib Ahmed is interested to know like how much time is required for preparation of these markers.
That's why I was telling that it is not a 1 year jobs or 2 years jobs. It's a difficult task. We need to have a lots of researchers, whoever is dedicated for this particular biomarker discovery, and they need to be together in hands and they need to develop. Then the time will be like it will reduce the time. Otherwise, normally if you go throughout this procedure, it will take more than 10 years because each like development of the marker in a laboratory is will take a like 10 years or 12 years, then we need to go for the clinical trial, and clinical trial is having a different phases: phase 1, phase 2, phase 3. Each phase will require for 4 to 5 years. So it's not like that there is a fixed time. It's required more manpower, so it will be easy and time will be less.
Thank you, sir. Our next question is for our resource person, Madam Payal Chakraborty. One of our student wants to know, she is interested, like, to know how to go for cancer research in future as a student of zoology, as it is her dream to do research in cancer. She is Minalini Bhattacharya, third semester, zoology honors.
Yeah, thank you. Hi, Minalini. There is no problem to go for cancer research if you are from a different field. It's actually a plus point. You can think freshly, and your expertise can be more helpful to fix a problem in cancer research also. So in Mizoram University, it's happening. Obviously, it's frequent that from other departments, students also can do research in cancer. So it's not a problem.
I hope, Nalini, you got your answer. And one more question we will take, like, ma'am, is why do people with the same cancer get different treatment and have different problems? This is
Yeah.
from Shubhito Sarkar.
Okay. Harshit, actually, what happened, as we all know, that our genome sequence is different individual to individual at 0.1%. And this 0.1% is making so much difference that one patient can have maybe mutation in this gene. But if you treat with one drug, then it will not helpful because maybe his immune system is not that much strong that it can take the toxicity of the drugs. So it happens man to man, person to person is always varies as our genetic makeup is different to human to human. Is it okay?
Yeah. Thank you, ma'am. Now over to Ma'am Shushmita for the vote of thanks.
Thank you, ma'am. Actually, there's a few more questions in the chat box. One
Yeah.
question is from Daulat Bhaumi. Can gene therapy or gene engineering can be a good option for the treatment of cancer?
Yeah, actually, the it's going on. The gene therapy is or genetic engineering, like a different CRISPR technique, is people are using for different treatment of the cancer. But it's too much expensive, and like in our country, it's underdeveloping country, it will be very difficult to do the gene therapy, and as well as if your like genomics is supporting or immunology or like immunity you're supporting, then only you can go for the gene therapy, because every time all the patients will not consider for the gene therapy because they will have a different genomic structure. So if you do the gene therapy, like a particular gene targeting a particular gene, like if you just I'll give an example, like most of the colon cancer and gastric cancer patients will not get the gene therapy because, like, if you having a particular gene EGFR, that is growth factor genes, that gene therapy is well established in western country. In India, in Tata Memorial, they are doing the EGFR targeted therapy. But if your genome has KRAS mutation, like a kinase mutation, it's a RAS mutation, that particular mutation, if it you have, so you will not consider for the gene therapy, for EGFR therapy, then it will be the severe condition for you. So that's why patient selection is more important for this kind of therapy. Yeah.
Okay, sir. Okay, sir. Last question from the student side. What is the main reason of uterine cancer and how this uterine cancer can be detected early?
Uterus cancer actually means uterus cancer main reason means it will be the genetic factor main things. then the lifestyle habits. Smoking is the major factor for all kind of cancer. First things, every time if you quit smoking, you will reduce 50% chances for the cancer.
Yeah.
Smoking, tobacco habit is the major reason for any kind of cancer. Uterus cancer, one of them for the female, as well as if you want to detect uterus cancer, you can detect by different cancer marker like CA 19-9, CA, and as well as you can go for the ultrasounds like deep ultrasounds, as well as a different kind of 64 slice CT. All these things you can do it. Then you can detect uterus cancer. There is no like a early detection marker for uterus cancer till now. Only the tumor markers are there that can be detected, but it will not be that much confirmatory. Confirmatory means you need to go for the deep ultrasound, or otherwise you can go for the 64 slice CT
Yeah,
scan.
so it's urinary tract infection. If you're having, then you are more is prone to get the urinary bladder cancer also. So we need to avoid the infection. So we need to maintain our hygiene, especially, and then yeah, lifestyle habits also. It's heavy. It's a very main factor to control it. But we cannot have any early marker.
Okay. Thank you so much, Sharon ma'am. So with this, I'll follow the instruction or the given by the ma'am Roomki. So a warm and cherished good afternoon and good morning to the resource person and all the participants. Uh, Honorable resource person, Dr. Payal Chakraborty ma'am, Dr. Shobik Ghatak sir, Reverend Principal Father Dr. Benike John CSC, Principal Holy Cross College, Agartala, respected Vice Principal Father and Administrator Father Joe Paul CSC, esteemed faculty members of the Holy Cross College, my dear participants, and everyone present here. It's a matter of privilege for me that I'm here. I'm proposing a vote of thanks for this program. I, Dr. Shushmita Shaha, on behalf of Department of Human Physiology, Holy Cross College, Agartala, first of all, my sincere thanks to Almighty God for making today's event a grand success. My heartfelt thanks to the respected resource person for accepting our request just to deliver your that wonderful journey of research through this online cancer awareness workshop. Thank you, sir and ma'am. We are really very grateful to both of you as you have spent precious time and you have given the very enlightened the young minds with this with your thoughts and experiences. And the both the session was really very informative. I hope all the students learned lots of things from both of you, sir and ma'am. Thank you so much once again. With deep sense of appreciation, I would like to express my cordial thanks to our respected principal father, Dr. Benike John CSC, being the catalyst who actually inspired us to do our best and stand there as a pillar of power. Thank you, Father, for sharing your precious time with us. Today we have started at 2 p.m. Till now, you are there. So from this, you are managing time from your busy schedule. So thanks a lot. Thank you so much, Father, for being with us. My heartfelt thanks to the Vice Principal Father, Joe Paul, CSC, for all kind of support and inspiration for organizing this awareness program. Here, I would like to mention my sincere sense of gratitude to the respected faculty members, especially Dr. Shomnath Kaur, Assistant Professor, Department of Botany, and Dr. Rumkinath Sen, HOD, Department of Zoology, Holy Cross College, Madam Shongita Chakraborty, Assistant Professor, Holy Cross College, for your all kind of support and presence throughout this online cancer awareness workshop. Now, A big thanks to Sir Gines Augustine for his efforts towards this coordination of today's program and for your support, for your presence, sir. Now, I would like to extend my generous thanks to all the faculty members of the Department of Human Physiology, Holy Cross College, including our new member, Kanak Chakraborty, for all of your untiring efforts to successfully organize this awareness program. Last but not the least, I am very much thankful to all the participants for your active participation. Thank you, everyone. I hope all the participants you have learned lots of things and little bit of idea. Now we can say you gain lots of idea and regarding the cancer awareness. So with this, uh, I would like to wind up today's session. I'll share the feedback link once again in the WhatsApp group, as well as I'll share the link into the respective email, those who have registered for the to attending this online cancer awareness program. Thank you, everyone. Have a nice day.
Thank
Thank you,
you.
ma'am.
Bye.
Thank you, Father. Thank you.
Thank you, Father.